Debido a que están por todo el mundo, o tal vez en una costa que produce esta estructura, que se parece a un llamado virus del libro de texto. Los supuestos virus vistos aquí se agrandan tal como lo imaginamos. Reproducir, reproducir, y me doy cuenta de que no va bien. No están sobreviviendo, y fui capaz de aislar estructuras de estas, y ver la misma composición bioquímica cada vez. Así que tenía algo y vi la misma proteína. Repetí el aislamiento cientos de veces, y con cada vez tiene el mismo ácido nucleico, la misma bioquímica. Había aislado algo y entonces fui capaz de mostrar pruebas suficientes de que son siempre idénticamente compuestos. A continuación vemos estas partículas que se supone que son el virus del sarampión, se nos muestran, y dicen que son virus. Aquí vemos un par de piezas pequeñas con apariencia circular, esférica, pero luego grandes pedazos aquí, y estas estructuras se nos presentan como virus. Nunca han sido vistos en humanos, nunca en animales, nunca en líquidos, en las membranas mucosas, en saliva, en sangre, en orina, nunca. Todo lo que se nos muestra como virus es falso. Son productos típicos de descomposición tisular de tubos de ensayo, con la que trabajamos, en la que intentas, o creer que puedes, estudiar la vida, que se remonta a Virchow. Era un patólogo que creía que mirando materia decaída de tejido en descomposición, de alguna manera podría aprender sobre la vida, y eso es exactamente lo que estamos haciendo hoy. Tomamos tejido de organismos como el tejido renal de mono que dura más tiempo en el tubo de ensayo, al menos eso es lo que les gusta decir, pero solo viven un poco más porque los rodeamos con suero de sangre tomado de un feto de vaca en el útero. El suero es sangre sin componentes sólidos. Si lo tomamos de vacas adultas o mamíferos crecidos sin células madres sanguíneas, el tejido moriría inmediatamente, pero con suero fetal permanece vivo por un tiempo y no se desintegra tan rápido, y estos signos de decaimiento en estas imágenes son un fenómeno específico único in vitro del tubo de ensayo que nunca se ha visto en la vida real in vivo. Tan rápidamente que pasamos de ser creyentes del virus hasta el fin de la virología y con el WIR. Estas partículas, aquí, se nos muestran como virus, pero, ¿qué son realmente? Toman tejido descompuesto, aíslan este tejido de todo alrededor del núcleo que aún puede sobrevivir, y partes del tejido desarrollan dedos pequeños, picos, o coronas, muy rápidamente, como una ameba que quiere reconstruir el tejido de nuevo. Es por eso que todos los que trabajan con estos cultivos celulares en tubos de ensayo tienen que separarlos constantemente una y otra vez. Estos trozos de tejido se malinterpretan como células, y estos pequeños dedos, con los que se arrastra para tratar de reconstruir el tejido. Estos se llaman las vellosidades, de las cuales una sección transversal en el microscopio electrónico debajo se nos presenta como virus. Es así de simple, es hocus pocus, truco de magia. Vemos que los virólogos, más tarde vamos a aprender por qué, son completamente anticientíficos porque nunca en ningún nivel de su trabajo hacen o documentan experimentos de control. Por ejemplo, podrían haber cortado trozos en el borde, o en el medio. En el medio, el diámetro parece grande, en el borde es más pequeño como este, y esto, y si estoy fuera de los trozos de tejido, entonces nada aparecería, y los que están en el borde son las únicas fotos que nos están presentando constantemente.
Today is the 6th August 2020, and I'm really happy to have Frederick Farway here with us. Frederick pursued his PhD in the lab of Michel Pactel, where he studied oncoprotein signaling in endoexosomal pathway, and he had pioneered a novel live imaging approach to study the dynamics of exosome release at single-cell level. He then moved to the lab of Grasa Raposo, where he established a novel zebrafish model system to track endogenous exosomes in vivo by live microscopy together with Guillaume van Niel. So currently he works in the now independent group of Guillaume van Niel to unravel the regulatory dynamics of exosome secretion and developing tools to further explore the pathophysiological roles of exosomes in vivo. So today he's going to share with us the live imaging approaches to explore exosome biology in vitro and in vivo. So please welcome Frederick.
Today I want to show you some of our recently published work on the tracking of EVs in vitro first. That's the first part of my talk, and also in vivo, that will be the second part of my talk. And so this work was performed in the labs of Michiel Bechtel in Amsterdam during my PhD. Subsequently, as Carolina just mentioned, in the lab of Grâce Repose at Institut Curie, and currently in the lab of Guillaume van Niel at IPNP in Paris. So it all started in Amsterdam around 2012 during my PhD in the lab of Michiel, when we were wondering, in fact, how we could possibly capture the dynamics of exosome release from cells. And the reason why we know so little about the dynamics of exosome secretion is that most isolation techniques, especially back then, require a large amount of cells that are cultured over a long time periods. And apart from the fact that this is, of course, a bulk approach, it also brings along the risk of contamination with cell debris and other extracellular vesicle subtypes that you might or might not be interested in. But most of all, these techniques, being post-secretion isolation techniques, do not allow to capture the dynamics of exosome release. So ideally, you would use a technique that allows for the direct real-time analysis of exosome secretion from at a single cell level, and also allows you to distinguish exosome release from microvesicle budding from the plasma membrane, as you can, for instance, see here in this scanning electron micrograph. So the approach that we came up with was actually borrowed from the neuroscience fields, and it was to develop and use a live exosome reporter called CD63 fluorine. Now, fluorine is basically a pH-sensitive variant of GFP that we then fuse to the first extracellular loop of CD63. And consequently, as long as CD63 fluorine is in the acidic lumen of an MVB, it stays quenched, but it becomes fluorescent upon fusion of this MVB with the plasma membrane, which results in a burst of fluorescence. And indeed, when we transfect this CD63 fluorine in cells, as you can see on the right, it traffics nicely to MVBs, and we also localize it to exosomes isolated from the extracellular supernatant. So this is, of course, very nice, but does it also work? And the answer is yes. So here you see a video of a cervix carcinoma cell that is actually very active in secretion. You see all these puffs here, these fluorescent flashes that correspond to MVBs that fuse with the plasma membrane. And since we're using turf here, what you're looking at is, in fact, the basal side of the plasma membrane facing the cover slip with a penetration depth of more or less 100 nanometers. So, of course, when we first saw this, we were super excited. But then we started to wonder, how can we actually be sure that indeed we're looking at MVBs that fuse and not, for instance, lysosomes or other transport vesicles that might also contain a small amount of CD63 fluorine and can also fuse with the plasma membrane. So to answer this question, the approach we took was live correlative light electron microscopy, or live CLEM. And this technique, basically, we combine dynamic live imaging data that usually has a low resolution or a relatively low resolution, that we combine then with electron microscopy analysis that then gives you the ultrastructural information. But as a downside has that it is static. So the way to combine these two types of information is, in fact, a bit similar to this last painting of Van Gogh called The Tree Roots. And this painting was, in fact, painted at an unknown location until recently, which means 130 years afterwards. Van Gogh expert found a postcard that looked oddly familiar to him. And then, indeed, by combining these two images, they saw a very nice overlap, a very nice correlation. And this is then how they identified the approximate location from where Van Gogh painted this painting. So CLEM is a rather similar process, except that it doesn't take us 130 years, luckily. So the first time when we had to optimize this technique, it took us maybe around 2 months, and now we can actually do it much faster now that we have optimized all parameters. So in short, what we did was we first acquired the fusion event by spinning this microscopy in this case. So directly after we observed the fusion event, we added a mix of PFA glut, processed these cells for electron microscopy analysis. And then, so by overlaying the two types of information, you can find back the spot on your section. And then exactly at the location of this fusion event, we created like a virtual Z-stack by tilting the section. And that allowed us then to reconstruct the fusion site in 3D by electron tomography, showing indeed a very nice MVB-PM fusion profile by electron tomography. So now that we know that indeed these fluorescent flashes correlate with MVBs that fuse with the plasma membrane, we can now start to further explore different stimuli and inhibitors. And we do so regularly by using this setup that you see here. So this is your imaging chamber that is on the constant perfusion. And then there's a superfusion barrel here that allows you to add stimuli or potential inhibitors very closely to your cells. And this setup then also allows you to image different cells before and after adding your stimulation. So by doing so, we, for instance, identified histamine as a trigger for exosome release in HeLa cells. And what you see here are total projection of fusion events during 1 minute time lapse that are projected as yellow dots here on the blue background of the cell. So here you see the control condition, and here you see the exact same cell, but after addition of histamine. And you can clearly see that there's an increase in fusion activity. So typically we then collect and record around 10 to 15 cells and can measure their individual responses to the stimuli or the inhibitor. And indeed, what you can see here that histamine significantly boosts the secretion of exosomes from HeLa cells. One other thing that is important to note here is that, in fact, there appear to be two levels of secretion. There's a constitutive, continuous level of secretion in HeLa cells, but we can also augment secretion or induce maybe even a subtype of vesicles that are secreted by adding histamine. So we don't know yet if in deals that we get with adding. This would be very interesting to look at, but we haven't had the time actually to do that. So then we can use this approach not only to look at triggers, but also, for instance, to identify snare proteins that mediate the actual fusion of MVBs with the plasma membrane. And doing so, we implicated SNAP23 and syntaxin-4 by using siRNAs. And in addition, also for SNAP23, we used a wild type and a dominant negative construct of SNAP23. So the dominant negative construct lacks the last 9 amino acids at the C terminus. And as you can see, this results in a stark decrease in fusion activity in these cells. And as you might expect, so inhibiting in this way with the snare protein results in a quite general block of exosome release, as we also observed a decrease for the release of CD81 and CD9 as measured by Western blot. Then using a specific set of inhibitors for G-alpha and PKC, we were able to further unravel the downstream signaling pathway that leads up to exosome release. But then to close the loop between histamine and SNAP23 function, we performed phosphoproteomics. And we did this on cells stimulated for 1 minute with histamine. And interestingly, this revealed a very specific phosphorylation site, 0910, that was highly phosphorylated after 1 minute of histamine stimulation. Now, interestingly, we created, of course, a mutant for this phosphorylation site. And what we saw was that it seemed to be quite specific. So in contrast to this general block with the dominant negative SNAP23, using this point mutation, we see a strong decrease for CD63 that is not there for CD81 and also not that much for CD9. And we also confirmed this with live imaging data. So this is quite interesting, as it might suggest that there's a certain level of regulation here on the secretion of MVBs at the level of MVB subpopulation. So this is also a very interesting phenomenon that we observed. Then the fact that these bursts of fluorescence, they correlate precisely with the site of fusion of an MVB with the plasma membrane. And this also gives us then the opportunity to see what other proteins or markers are there right before or right at the moment of fusion. So in other words, we can address what these MVBs look like right at the moment of fusion, and by doing so unravel their molecular identity. We described this approach already in our recent Nature Protocols article by taking the example of CD63 fluorine co-transfected with an other tetraspanin tag to RFP. But of course, nothing prevents you to also do this with other proteins than tetraspanins. So for instance, here I co-transfected CD63 fluorine with Rep5 tagged with RFP. And as you know, Rep5 is an early endosomal marker. And what you can appreciate from this imaging is that the fusion spots are only green, they're not yellow, meaning that Rep5 is absent, is not present on the MVBs at the moment that they fuse with the plasma membrane, which makes sense, Rep5 being an early endosomal marker and MVBs being late endosomes. Rep7, however, is a different story. Rep7, of course, is a late endosomal marker that inside a cell overlaps significantly with CD63 across cell types. And it has also been implicated already in exosome biogenesis before. To our surprise, however, again, we did not observe Rep7 at the site of fusion. Then, by contrast, the late endosome-associated Rep27, A and B, that have been described by the group of Clotilde Théry, Matthias Ostrowski in 2010, to be responsible for trafficking towards and docking of MVBs to the plasma membrane, we could clearly see that this Rep27 was there. So in fact, if you look carefully, you see here a red vesicle that at a certain point turns yellow when it's actually fusing with the plasma membrane. So what is actually happening here? So you might be familiar with the fact that usually if you go from early endosomes to late endosomes, you need a switch from Rep5 to Rep7. And this data also then now suggests that in order for an MVB to fuse, you need to lose Rep7 that need to be replaced with Rep27. So we're currently looking into how this might be regulated by the cell. And I will actually show more on this during the upcoming EV Imaging in Vivo workshop coming September in Paris. So then finally, to conclude this in vitro part, I just want to briefly highlight the recent story by Asang et al. In the group of Alyssa Weaver that have also worked already for many years on CD63 fluorine in independent research line. They have recently published an updated version of CD63 fluorine that they then linked to fuse to M. scarlet in purple. So then you have a dually labeled CD63 fluorine. And by doing so, what is very nice here is that you can also study the uptake. So exosomes that are here deposited are mostly white, yellowish. But then after uptake by the recipient cell, you will see that because of the acidification of the compartments, you lose the green color, making the exosomes fluoresce only in MS scarlet. So this is a very nice approach to also study uptake using a fluorine approach. Then the last technical notes before we move on to the in vivo part. So in the protocol that we published on the use of CD63 fluorine, we also describe an assisted counting tool, as we call it, as you can see active here on the left. And the reason why we developed this tool is in fact that not all events are clearly events. So of course you can also have neutral vesicles that approach the membrane and then return back into the interior of the cell. And in addition to this, there might also be events like kiss and run events that maybe do not really release exosomes. So we developed this tool together with Philip Dunn here at the Neurimac imaging platform at the IPNP. And we set up rules to determine what is a fusion event and what is likely not. So it allows you then to click on the event and ask the program, ImageJ, in fact, to determine for you whether the event is a real fusion event or not. So we compare this also on the same imaging data set, the manual analysis, the old-fashioned way, let's say, with the analysis using the plugin. And indeed, you can see that also the plugin is able to pick up physiological differences. You see also that it's a bit more strict, but what it brings you is actually a more standardized and more objective way to analyze your CD63 fluorine live cell imaging data. And also one last thing is that the construct CD63, CD81, and CD9 both tag with fluorine or Fuji, and Fuji is the red variant of a pH-sensitive probe. They're all available via AdGene. So if you're interested, you can request them there. So some very generalized conclusions for the first part. What we've seen is that exosome release is in fact a dynamic process that can both be continuous but also induced. There's an impact by the cell's environment on the release activity of exosomes by cells. And then furthermore, it's governed and modulated by more cell-intrinsic processes. So follow-up questions that this provokes and that I actually do not necessarily have the answer to are, for instance, how do even standard cell culture conditions impact quantities of EVs that are released, but also their content? And more related to the release dynamics, how continuous versus induced secretion might affect efficient content delivery to receiving cells. Now, of course, this stresses the need for complementary in vivo model systems that allow us to validate in vitro findings and also to explore exosome communication in a more physiological context. So with that, we've arrived at part 2, which is the tracking of exosomes in vivo. So to track exosomes in vivo, basically what we've done is we've transposed the CD63 fluorine in vitro to the zebrafish embryo model system. But why zebrafish? Zebrafish embryos represent a naturally transparent vertebrate model that has a high genetic amenability, a fast reproduction rate, and also a very well-defined vasculature. And due to this transparency, it also allows for imaging with a very high spatial temporal resolution, which is ideal if you want to ask a cell biological question at an in vivo level. So using this model now, CD63 fluorine in zebrafish, allows me, first of all, to express CD63 fluorine in a tissue-specific manner. And I can do so, for instance, by injecting it in a cell-specific manner or by making use of cell-specific or tissue-specific promoters. And what I will show you during this, the remainder of this talk is that I will mostly use the cell-specific injection. For this, we target the yolk syncytial layer, and as the name suggests, it is a syncytium that covers the yolk of the embryo. And we found that this is a highly active secretory layer during development. So by expressing it in the yolk, we could then also confirm that with electron microscopy, that indeed this yolk syncytial layer is basically generating loads of small extracellular vesicles in the size range of exosomes that are also labeling highly positive for anti-GFP immunogold. And this anti-GFP can recognize the fluorine moiety of CD63. But then after this tissue-specific expression, this model system also allows us to follow exosomes after their secretion and follow them throughout the embryo. For instance, we have observed these YSL-derived EVs here in a vein posterior to the head. I hope you can see it. So you will see these very small fluorescent dots moving through the blood flow. Then you see these black holes, which represent red blood cells. And also here, again, using in situ electron microscopy on the vasculature of the fish, we could confirm the presence of small extracellular vesicles in the size range of exosomes, again labeling highly positive for anti-GFP immunogold. Now, apart from the blood, we can also observe these exosomes in the interstitial fluid. So in this case, you're looking at the interstitial fluid that is covering the muscle cells that are very highly abundant in this particular area. And apart from the interstitial fluid, we also found these exosomes in the neural tube. Then finally, here we're looking at the tail end of the fish, the CVP area, the caudal vein plexus. So you see the caudal artery, the CVP, and the cardinal vein. And also here you see all these exosomes transported via the blood to their target cells. Basically, the conclusion here is that exosomes are everywhere. Then, apart from tissue-specific expression and the visualization of their trafficking, we can also use this model system to identify target cells and molecular uptake mechanisms. And this video here is just to give you an impression of the resolution we can reach. So the vasculature is in red here. You see some macrophages crawling down the endothelium, and in green you see the exosomes that interact both with the endothelial cells and macrophages. So this is more of a close-up, a zoom-in on the macrophage in the CVP, outlined here in this white dashed line. Then you will see a vesicle arriving here at this place, and now I will restart the video, and you will see that in fact this macrophage scratches off the exosome that was arrested on the endothelium and internalizes it. Now then, to show you just that it's not a one-time occasion, but actually it seems more like a full-time job for these poor macrophages, we're looking here at an embryo, a fish line that has the macrophages labeled with M. cherry, and you can indeed see that these macrophages are super active in taking up all these extracellular vesicles that they find around them in green. But what about the endothelium itself? Here you can see a real-time stream of the cardinal vein. You see the vessel over here, more or less. And what you can clearly observe here is that some of the particles in the blood flow slow down and are arrested along the endothelium. So you see 2 occasions marked by 1 and 2. So they are arrested in the cardinal vein on the surface. But then to validate whether they also taken up, we have an issue, of course, because the compartments in endothelial cells where exosomes might end up in acidify very fast, and the fluorescence of these EVs will therefore be quenched, which makes it hard to appreciate the exosome turnover that is potentially happening in these cells. Now, to prevent this masking effect, we inhibited the proton pump using bifidobacterium, and indeed we could confirm the presence of significant quantities of EVs in the endothelium of zebrafish. So indeed we can see that the endothelium takes up YSL-derived EVs. And you can see here also in this time lapse that actually this internalization pathway is highly active. So the imaging starts more or less at the point that bifidobacterium has completely neutralized all the acidic compartments. And what you see here is that already in 30 minutes, there's already a significant accumulation of EVs happening. Now, strikingly, as I told you, the interstitial fluid that's also full of YSL-derived EVs is completely covering muscle cells. So basically, they're completely drenched, bathed in a soup of EVs that are present in the interstitial fluid. However, I looked very hard. I used very high amounts of bifidobacterium, longer time periods, but I never observed any fluorescence inside these muscle cells. So this suggests that endothelial cells that you see here express somehow a specific receptor that allows them to bind and internalize these YSL-derived EVs. And as a matter of fact, indeed, this specific area of the vasculature, the CVP, is particularly enriched in scavenger endothelial cells that, as the name implies, express scavenger receptors that anisotose macromolecules in a dynamic-dependent way. So to validate this, we use dextran sulfate, which is a broad inhibitor of class A scavenger receptors, and basically competitively binds to the scavenger receptors present on this scavenger endothelium. And indeed, we observed, as you can see here, by imaging, a significant reduction in the uptake, as you can also appreciate from this quantification graph here. And we observed a similar decrease in uptake by using a specific inhibitor for dynamin called pyrimidine. Also here you see a stark decrease in uptake by these endothelial cells. To study the fate of these YSL-derived EVs in the target cells in the CVP, those endothelial cells. So here you see an electron micrograph of here's a lumen of the blood vessel. You see a small red blood cell over here, an endothelial cell over here, and then if you zoom in on this area here to the right, you can indeed see CD63 fluorine labeling in this electron-dense compartment, suggesting that after uptake, actually, they enter degradative compartments. This is actually highly comparable to the back-to-back publication of Vincent Yen in the group of Jackie Goods in Strasbourg. What they showed was that in accordance with this, tumor-derived EVs that are labeled and injected in the zebrafish blood flow also end up in the same endothelial target cell population and seem to also end up in endolysosomes. So now, if these YSL EVs do not seem to be functionally transferring any cargo, but instead are quickly degraded in a target cell, what could their function be? And for this, we first wanted to focus on the content of these YSL-derived EVs. And for this, we took an ex vivo proteomic approach. And to do so, we first dissociated the embryos that were expressing CD63 fluorine in the YSL. Then we isolated all EVs by differential ultracentrifugation. So of course, then you end up with YSL-derived EVs, but also EVs that are potentially generated by other tissues in the zebrafish embryo. And then to enrich specifically for the YSL-derived embryos, we use beads coated with anti-GFP to really IP the YSL-derived EVs. This then we sent for proteomics and compared it to a background of fish processed in the same way, but not expressing CD63 fluorine. So then you get a list of enriched proteins. And the number one most enriched protein then was Alex, which was 34 times enriched in YSL-derived EVs. And there are even some proteins that we exclusively found in the YSL-derived EVs, such as synthenin and THG101. Or also Rep7. Apart from these proteins, we also found Chim4C, for instance, CD81, CD63, flotilin, cation-independent mannose 6-phosphate receptor. And all these proteins together then form some sort of late endosomal signature, and also provide, in fact, a clue towards the in vivo biosynthetic pathways for these exosomes, namely the Alex-synthenin pathway. So a lot of our data, in fact, including our electron microscopy, then points towards the MVBs as a potential origin for at least part of these YSL-derived EVs, suggesting that they are bona fide exosomes. So then we looked at the GO-term enrichment for all these EVs, and we found a lot of terms, in fact, linked to metabolism. And this could indicate a role for these EVs in trophic support of the developing embryo, and maybe in particular the CVP area. This was also previously suggested, although in a different setting and demonstrated by those two papers. So to further follow up on this lead, we then opted to interfere with the biogenesis of these EVs in the YSL by using, so actually based on a hint that we got from proteomics data that showed that the Alex-synthenin machinery was involved in the generation of these YSL EVs. So then we used morpholinos, which are the equivalent of more or less of siRNA approach to knock down synthenin specifically in the YSL. And indeed, as you can see here, we were able to significantly reduce the release of fluorescent exosomes from the YSL that we could rescue actually by using a synthenin construct that was insensitive to this for the target site, mutated for the target site. Then by looking closely at the size and surface area of the recipient tissue in these embryos, we found a significant decrease in the size and the growth of the CVP in zebrafish blocked for the release of these YSL-derived EVs. And indeed, further analysis then revealed that this difference was mostly due to a defect or a decrease, a slowing down in proliferation, whereas the nuclear density, so that means that the amount of nuclei per surface area remained the same. And this really suggests indeed that these EVs coming from the YSL, which in fact contains all nutrients, have a role in trophic support of the growing embryo. So to now finally conclude, what I've shown you is that you can use CD63 fluorine expression in vitro to quantify exosome secretion at single cell level, identify exogenous stimuli, different unravel signaling pathways, and also study cell intrinsic modulators of secretion. Then by using it in a zebrafish embryo model system, it also allows you to track the dynamics of these exosomes after their release, their behavior and biodistribution at single vesicle resolution. Interestingly also, what you're able to do then is to do proteomic profiling, ex vivo proteomics on tissue-specific EVs by pulling them down from the general population with anti-GFP beads. And finally, it allows you then to study the release and uptake mechanisms and their function in vivo in the physiological context. So Basically, to summarize this last part in one sentence, is that I found that physiological EVs from the YSL target and are rapidly degraded by liver-like scavenger cells, seemingly without having a clear message function, but rather seem to function in trophic support. So then finally, I'd like to acknowledge that the different groups, the group of Guillaume van Niel, of course, also the Exosome Research Group in Amsterdam, in particular Michiel and Maarten, who also significantly contributed to the JCB and Nature Protocols, the group of gastropods at Institut Curie, the group of Filippo D'Obena, that is now at Institut Imagine, and also, of course, our collaborators in Strasbourg, Jackie and Vincent, and Philippe Herboma at Institut Pasteur. Then finally, I'd like to point your attention also to this, as I mentioned already, this workshop that we're organizing that will take place 10th and 11th of September, so next month. And so although the subscription is closed, of course, we are considering now ways to make part of the content available virtually, and we will update the website in case this is indeed possible. So thank you for your attention.
Thank you very much, Fred. It was great, beautiful images and so on. So to run the questions here, we ask you to put your question into the chat box, and then I will convey those. No, you will be. I will give you space, and then you will need to unmute yourself. So Min, please unmute yourself and ask your question.
Hello.
Yeah, we hear you.
Hi, thank you. It's very nice talking. I really like the data you have, especially the video. Maybe I missed it. What type of EV did you inject in the embryo? Is that zebrafish EVs? And if you use the EVs from a different species, do they have a different fate, like more phagocytosis? And if you EV from different cell type, do they end up in different places? And did EV get through the endothelium by transcytosis? Do they get through the blood-brain barrier? Sorry, it's a few questions.
Yeah, that's a lot of questions. So to answer the first question, in fact, you can use both approaches. What we did was mostly just expressing CD63 fluorine in this yolk syncytial layer. So in this case, you label endogenous exosomes. In the back-to-back paper by the group of Jackie Goods, they indeed also, so this is also possible, they isolated exosomes in vitro, labeled them with membrite, and then injected them in the vasculature in the blood circulation of the fish. So there are already two approaches that you could take. And indeed, what would be very interesting then is to see whether these exosomes have different targets. What we've seen so far is that these tumor EVs actually seem to have the same target as the endogenous EVs that come from the YSL. Then regarding the transcytosis, so we have some indications that this might happen indeed via the endothelium, but we have no solid proof as of yet. But this is indeed, it's interesting. I think it has been described also, although not in zebrafish yet. But this is definitely an interesting point on the to-do list, let's say. Yeah.
I'll follow up with that. Do you think you can make vesicles target specific cells in your system? So exclusively macrophages or exclusively endothelium, for example?
Yeah, I think so. So if you look at the work of Hoshino et al., it seems that by using different types of integrins, you can target different cell types. So far, the exosomes that I've looked at all go to the liver, which is the CVP, which is an area that in the embryo actually highly resembles the liver in mammals. Mm-hmm. But I think that if somehow you can prevent their targeting to the liver, they might go to other places. So yeah, the question there is indeed does this uptake, this scavenging by the scavenger endothelial cells, does it prevent the targeting of these exosomes to other cell types that they would otherwise go to? So we haven't looked at this yet, but it's clearly something we want to do, for instance, also by using different tetraspanins. But I think that trying exosomes isolating from different cells, expressing different pairs of integrins could also be a very nice approach. And finally, if you look at the drug targeting and delivery field, they're also able to, most often what they see is also that most liposomes target the liver, but they are in fact also able to minimize this a bit and, for instance, target more cells in the blood-brain barrier. But this could also be very interesting indeed.
I thought that you saw some EVs that go to other organs. I imagine that those are the endogenous EV, right? If they come from if they are endogenous EVs, then they should not be taken up so much by the liver, isn't it?
I would not consider it as foreign, but just at a very specific tissue that is very important in it's a very highly secretory layer, and it's very important in delivering the nutrients that are present in the yolk to the developing embryo. Of course, they need all lipids, etc., protein, amino acids, etc., to basically to build new cells. And in that sense, the CVP makes sense to me as a target tissue.
Thank you. I'll let Jason unmute himself and ask the question. Next question.
Yeah, great.
Please say where you're from, actually. I forget to ask people where you're from. So you tell us where you're from.
Melbourne,
even if
Australia.
I know, I won't say.
Okay. Melbourne, Australia. Really nice talk and great imaging, obviously. So can you tell me, has anyone shown, that trophic support sort of hypothesis through the, endolysosomal system? So you're suggesting that these EVs are being taken up, they're going through the endolysosomal system. How are they actually providing that trophic support? What do you think is happening there?
Let's see, I will just stop my PowerPoint presentation. So there was this study in eLife in 2016, the one that I referred to by Zhao et al. What they have shown actually is that in the context of a tumor, it's a prostate cancer, that the CAFs are induced to release exosomes, and these exosomes then contain different metabolites that then are transferred to the cancer cells and actually reprogram their metabolism and also are incorporated in the different pathways. So that is one way how this could be done. So apart from that, I'm not really aware of a lot of other studies that looked at that. But this is definitely very interesting. We see basically the bulk of exosomes that you inject usually in vivo goes to the liver. So this is a significant pathway. What it means remains to be further.
Your uptake experiments are really nice, though. They look very nice. And there's a lot of debate about when things get taken up. Do they provide a function to that cell that they take up? And why would you take something up just to degrade it? It doesn't make a whole lot of sense either. But, what is the actual support? Maybe it may be just membranes, lipids that they're supporting and things like that that you use.
So it could indeed be lipids. Of course, they certainly have lipids. So the yolk also contains a lot of vitologen, which can also be broken down and reused for the growth of recipient tissue. We also compared, actually Guillaume also used this analogy of like a protein bar, a power bar that is highly concentrated in nutrients and then delivered to a target cell that can then use it to generate new cells, basically.
Can I ask you quickly, so also, you show with CD63 really nicely that these EVs are traveling around. Have you used a different fluorine attached to a different tetraspan and seen that they travel differently? Or have you just done the CD63 so far?
Yeah, so far I've only done a quick test with a CD9 fluorine. So this was just one experiment, and they roughly also seem to target the same tissue. So also the CVP. So what might be actually different is, or when you might expect to see differences, maybe just to target another producing tissue, for instance, the muscle cells or the endothelial cells. That's what the ARN is doing, I think. So this could be very interesting. Yeah.
Cool.
Thank you. Aaron, please state where you're from, etc.
Yeah, so from the University of Bristol. So yeah, thank you very much for a really enjoyable talk, Frederick. So I've often thought, you touched on it in your talk, and I've often thought about the really exciting opportunity of placing your reporter under the control of a cell type specific promoter rather than the ubiquitous one. So first of all, I was kind of wondering how that was going. And then I was also just kind of further to that, thinking whether you might encounter issues relating to expression stability, and would a similar approach to Alyssa Weaver's lab, introducing that stabilizing mutation, allow you to get that step?
Yeah. So the tissue-specific promoters, indeed, I've made use mostly of the ubiquitous promoter. In the meantime, we also have developed, but it's not published yet, UAS reporter. So with a GAL4, you can cross it with a GAL4 line to express it in a specific tissue and actually generate stable lines. So and then the second part, compared to the related to the stability. Yeah, so that's a good point. We also came across this M153R mutation that was published for a fusion protein, stating that it increased the stability and the fluorescence. In our hands, but I think it's really cell type dependent. In our hands, we do not seem to have much of an increase in brightness, and also in our hands we can generate stable lines with the normal CD63 fluorine. But we are doing tests also now with this plasmid to further. Yeah, the analysis that we've done so far is a bit preliminary, but we think actually that also, for instance, regarding to the cleavage, this might also relate a lot to the expression level that you have in your target cell. And yeah, indeed, what we would like to do is really to put them side by side and see if there's indeed less cleavage. Because actually in the paper you can see that there are still some cleavage occurring even with the M153R mutation, but it could be significantly less. But yeah, so we are actually currently doing these experiments to put them really side by side. Yeah.
Brilliant. Sounds really positive.
We move to Eken and Ankor. Please state where you're from. So basically, he's asking, could the CD63 fluorine be used to study exosome biodistribution and secretion and uptake by a specific cell in more complicated organisms beyond zebrafish embryo?
Yeah.
Time courses and so on, yeah.
Yeah. So the reason why I chose to do it in zebrafish was for their, let's say, optical superiority compared to murine model systems. I do definitely think that that you can use CD63 fluorine in mice, but the imaging will be much harder. So usually you're more limited to the area that is really close to the skin. So you can do an imaging window as done in the group of Jacco van Reenen. So I think personally I would rather use them as complementary model, but use them to answer different types of questions. I think that really the high resolutive single vesicle tracking is really hard to do in other more advanced models in that sense.
Thank you. Then we move to Thak Pham. Please unmute yourself and ask a question.
Hi, Frederick. Thank you very much for a very nice talk.
And where are you from?
I'm from Singapore. So, yeah, I'm really interested in see the macrophage actively uptake the exosomes, and I just wonder why this uptake a lot. Yeah, it just secrete out and then uptake by macrophage. It is like kind of waste. And does macrophage uptake more or endothelial cell uptake more exosome?
So that's a good question. I actually don't know I haven't quantified really the amounts of uptake in macrophages versus endothelial cells. They do clearly, macrophages, they do clearly take up significant amounts of EVs. So the reason why they do it might actually be more linked. Also, again, if you do the analogy to the liver, so these macrophages would then be comparable to the Kupffer cells that actually also in mammalian embryos are from the same origin. So they originate from the yolk. And in this case, for the liver, it's known that these Kupffer cells also have, for instance, an immunological function. So it's more some sampling of what is present in the blood, and then, if necessary, to elicit an immune response. So that's something that could be happening here. It could be some sampling, but at least we can see that even though the macrophages take up per cell a significant amount. My guess would be that most uptake of the EVs coming from the YSL is done by the endothelial cells in the CDP, also because they have this very high, very fast turnover. You can actually see that, I didn't allude to that further, but you saw also that in the video. So this was not, the video of the macrophage was not with baflomycin. You can actually see that for some reason, the fluorescence of the EVs in the macrophages is still active, meaning that they are not in acidic compartments. So they might actually have a different, indeed have a different function and not be rapidly degraded as you see in the endothelial cells.
Thank you. So we move to Ananga Ghosh. Please state where you're from.
Yeah, so very nice talk. Thanks for the very nice talk. I'm from India. And I am actually very new to this field, so I am asking a very little bit not related to this topic, but a general question that whenever we do some knockdown or something to show that exosome production is less, always I've seen the papers also that people have shown that after doing a Western blot to show that the exosome production is less after doing the exosome purification. So is there any other way to show that really exosome production is less when you are doing some knockdown or some one?
So what I've done in the paper was just to use more like a correlative way to assess the amount of exosomes. So it's not absolute quantitative way, but I've just measured the fluorescence that can only come from EVs carrying the C6 fluorine in the blood flow. So I've just measured the intensity over the background and compared that in the different situations. So the other way would be more complicated, I guess, because, for instance, if you do the dissociation, you end up with a lot more exosomes than just the ones coming from the YSL. So unless you do an embryo-wide knockdown of exosome secretion, I think it will be hard to do it in another way.
Okay. Okay, so generally, like in many papers, I've seen that whenever people like if they are knocking down RAP27A or TSG101, so always they show that after exosome purification, they do a Western blot and so that these markers are less in the norm compared to the normal. So that is the general way to show, right?
Yeah. So in vitro, that is the standard approach that you would take. But I thought you were referring mostly to how to confirm it in vivo. I think that is that's
in vivo
much
also
harder. Yeah.
Yeah, so in vivo is much more complicated, but in vitro also that is the general accepted way, right?
Yeah.
Okay, thank you so much.
You're welcome.
So I actually have a question myself, unless there are more questions. So just put the questions out there. And I'm thinking more about subpopulation of vesicles that carry specific types of molecules. For example, we published last year that there is a subpopulation of vesicles that, at least from some cells, that contain mitochondrial inner membrane proteins and not outer membrane, inner membrane proteins. And we believe that's probably happening through mitophagy processes, which is something I'm not really very familiar with scientifically. Are you aware of any work, or have you done any work around other types of vesicles rather than the standard vesicles?
No, not really.
Subpopulations, specific
Yeah.
subpopulations of vesicles.
No, so apart from this, doing this IP on tissue-specific exomes, I've not looked yet much into subpopulations, but of course it is possible to use the different tetraspannins for that. And so the result that I showed you with this specific phospho mutation in SNP23 suggests indeed that like if you co-express both, of course you will see quite some extensive overlap. But if you look at the Western blot that I showed was actually for endogenous exosomes that were released, and then the imaging data obviously was done with overexpression. But we see some clear differences there, but we haven't gone really into much more detail there. Although it's super interesting. Yeah.
Thank you. Thank you very much. I give the word over to Carolina, and we really enjoyed this session.
Thank you so much, Jan, for mediating the question and answer. And Frederick, there's a wonderful presentation. Thanks so much for sharing your data with us. And I think,
pleasure.
Yeah, today we learned a lot about the CD63 pH lore and also the construct that can be used to visualize this EV secretion and fusion events. And there's also interesting that some vesicles seems to be uptake by macrophages and endothelial cells, but not the muscle cells. So there's quite a little bit of not a little bit, actually a lot of follow-up to do, which is going to be very interesting in the future. Yeah. Thank you for that. And I think with that, I'll wrap up the session.